So this is antibody-mediated immunity. When the B cells encounter an antigen, an unprocessed antigen, so that would be an antigen on the microbe or on the pathogen, um, it becomes activated. So the antigen itself binds to the receptor on the B cell that's specific to that antigen, and it activates. Now, B cells can also process antigens. They can actually be taken into the B cell, and then the B cell can act as a antigen presenting cell because they have MHC2s, the same as a macrophage or whatever. Um, so if the antigen ends up, if the microbe ends up inside the B cell, it will act as um, as an antigen presenting cell. Um, but what will happen is that when the B cell becomes activated, it undergoes a clonal selection and makes identical copies of itself. And again, there are memory cells that don't have anything to do with this round of infection, and there are the action cells, the activated cells that happen. Now, when a B cell, an activated B cell, the action part of that that's going to make the antibodies are called plasma cells. So the B cell morphs into what's called a plasma cell. So the B cells have receptors. They meet the microbe. They they activate. They can also be activated by the helper T cell. They can be co-stimulated. But no matter what, they end up producing memory cells and plasma cells, and the plasma cells secrete the antibodies. Now, antibodies are extracellular proteins that are made by the plasma cells, and to a lesser extent by B cells, that have specific functions. We'll talk about them in a minute. But it's important to realize that they are made on rough ER. They are just proteins, and rough ER is where proteins get made. And if you remember back to physiology and translation, transcription, protein synthesis, it takes endoplasmic reticulum with ribosomes on it to make these proteins for export, to be outside the cell. Before the B cell can actually start making a bunch of these antibodies, therefore, it needs to grow a whole bunch of endoplasmic reticulum. At that point, they're no longer B cells because they are B cells with this extra endoplasmic reticulum, so we call them plasma cells, as in endoplasmic reticulum. The same thing sort of happens when an immature hemocytoblast, is going to become a erythrocyte, a red blood cell, and they have to make a whole bunch of hemoglobin, which is a protein. The first thing they grow is endoplasmic reticulum so that they can function to make the hemoglobin. We call those cells reticulocytes because plasma of the endoplasmic reticulum was already taken by the B cells, the name, and the name, so they are called reticulocytes, as in endoplasmic reticulum. So what are antibodies? These are these extracellular proteins, and they combine with the antigenic determinant of that antigen that triggered its production. So it directly interacts with the antigenic determinant of the antigen that called for its production, that activated its specific B cell. And these proteins, the glycoproteins, and they become the globular, so they become immunoglobulins. I'm not really too worried that there's 4 polypeptide chains, heavy chains and light chains, and a hinge and a variable region and a constant region. But what's important is that there are 5 classes of them, and each class determines a different type of reaction. So so this one happens to be an IgG, immunoglobulin G class. And they're kind of Y-shaped, and they bind to the antigen over here, right? And the stem region sticks out. So this binds to the antigenic determinant of the antigen. A couple of different proteins, but you can see why they're globular proteins. So what do they do? By sticking to the antigen on the surface of the microbe, they basically neutralize the antigen. It means that the antigen can't interact with anything. It's like swarming. It's like when your friend is mouthing off in a bar, you surround him and you hustle him out of the bar to keep him from getting into trouble. Same sort of deal. Another thing they do, by sticking all over the outside of a bacteria especially, they immobilize that bacteria, which means they stop its ability to swim. They basically drag it down, and so that the macrophages can get to that bacteria and eat it. It can't swim away from the macrophage. And so it makes it easier to be caught because it's immobilized. It also can't spread as well because it's immobilized by the antibodies. Antibodies agglutinate and precipitate the antigens. Agglutinate is like it's like a chain gang. It's like a whole bunch of convicts being handcuffed together to a chain. They can't get away because they are all attached to one another, and that's in terms of cellular stuff. In terms of immunity, we call this agglutination. We also make it so they're not soluble, so they precipitate. Again, if they're not in solution, then they're more difficult to get away, and so phagocytosis happens. They are involved with enhancing phagocytosis by immobilizing and agglutinating, but they also provide a handle for the phagocyte to grab hold of. They kind of work with complement on that. And so they enhance phagocytosis by not only targeting the antigen of the microbe, but making it so that the phagocyte can actually grab hold and it can't get away while it's being engulfed. And then it activates complement. So back when we were talking about the non-specific, we talked about complement being a series of proteins that get injected into the cell membrane of the microbe and does a few things. One of the things it does is it enhances phagocytosis, opsonization. It makes it, opsonization literally means to make tasty. It means that the phagocytes will grab hold of it easier. They cause cytolysis, which is basically poking holes in the microbe so that the endoplasm of the microbe leaks out and the microbe dies, or at least is wounded. And complement also is one of the triggers for inflammation. And all of this is an example of positive feedback homeostasis, a cascade, as it were. Once you get the complement activated, then it activates more of it and more of it and more of it. One reaction triggers the next. There are a bunch of different pathways that make it happen. I'm not going to worry about the 3 different pathways and C3, but nevertheless, this is what happens. What it turns out to be is this, that this is the are a bunch of the complement proteins. They inject into the cell membrane the lipid bilayer of the cell membrane of the microbe. They form a ring which makes a hole. We call this ring the membrane attack complex. It makes a channel, and then everything inside will flow out. So basically the cell bursts, the microbe bursts. As these things come together, it triggers mast cells here, especially the 5A's, will trigger the mast cells to release histamine. And histamine is a potent vasodilator, which causes vasodilation, slows down blood, enhances emigration, causes inflammation. And remember, inflammation causes pain, redness, immobilization, swelling, and heat. And all of those revolve around vasodilation, which is caused by the histamine released by the mast cells, which has been told to release it by the complement protein. The other thing that it does is complement is sticky to the macrophage, so it enhances phagocytosis. These complement proteins mean that the microbe can't get away as it's being engulfed by the phagocyte. So in this way, the specific antibodies here enhance phagocytosis by activating the non-specific complement cascade, which so they work together on that. Now, these memory cells that are made make it much more readily available to make these antibodies to do this stuff. Oh, by the way, there are 5 types of antibodies, MADGE. I don't see a slide on that. I will see if I can find something, and I'll post it, probably with very limited commentary. I'll find it from a different textbook. What I'm going to talk about here is that when you are first exposed to a pathogen or to a microbe, then what happens is you get quite a few antibodies, and they come kind of in this 14-day time frame. And when you think about it, that's about how long you're sick for, a week and a half. You know, it kind of peaks out here at about 10 days. That you have the most antibodies at about this point, at about the 10-day point. And that's about when you're no longer contagious. That's when you are winning against the infection. It takes a while. It takes quite some time to get the titer, the number of these antibodies in circulation, up high enough. Because what's happening here is that you start out with relatively few B lymphocytes. They have to activate, then they have to clone, and then the clones have to get activated, and then they have to turn into plasma cells, and then they start pumping out antibodies, and the population is fairly small. So by the time you actually get quite a bit of antibody, of IgGs, it takes quite some time. And then they have to interact and agglutinate and precipitate and activate the complement, and it takes a while for you to win. But if you get a second exposure, these memory cells, there are a lot more of them. So what happens is the response comes sooner. The first exposure, the first meeting between a naive B cell and the antigen is more likely to happen. And when it happens, there's more of them to to do it, and so there's more of them to clone, so you end up with way more clones, way more memory cells, way more cells turning into plasma cells, and they all start pumping out antibodies at their normal rate, but there's so many of them that you get a lot of antibodies, and it happens really, really quickly. And you can see that this graph, the second exposure, has a huge number of antibodies that give you protection. So not only is it faster and happens sooner, but it's much, much stronger and gives you that much more protection. So this is why you tend to get sick on the first exposure, but the second exposure and subsequent exposures to a pathogen tend to not make you sick. You get exposed to the mumps here when you're a kid, either through the vaccination or because you had the mumps, and then the next time you encounter the mumps, you don't actually get the disease. You have a reaction to it and you fight it. This is the thing that excuse me. This is the thing that we're checking for with the tuberculosis skin test. We want to see if it inflames with a little bit of tuberculous antigen, and it means that if you get the inflammation, you've been exposed at least once. It doesn't mean that you have tuberculosis, but it means that you have antibodies against the tuberculosis, that you've been exposed to it, and that you've successfully fought it. It's a screening test because if you don't get this secondary response, you've never had the primary response, which means it's impossible for you to have had tuberculosis or to have it now. If you have the secondary response, that means you've been exposed and you may or may not have it, and so then further tests are done, like chest x-rays, etc., to see if you have tuberculosis or whether you successfully fought it when you were exposed. That's the basis of it. And you can go back and look at that the complement causes the immunity, or it causes the inflammation, and the complement is targeted by the antibody specific to that thing. What you don't want, and this is where the pathology comes in, is you want the cells to have self-recognition and self-tolerance, to not react to your own proteins. You want it to be able to recognize your own MHCs. In our thymus, any T cells that don't have self-recognition die, right? And self-tolerance occurs through negative selection, where any cell that recognizes self-peptides and is activated by them are killed either through deletion or by energy, which means that they're still alive. Obviously there's a spelling mistake in remain. They're alive, but they become unresponsive. So that's an important point. So the last video in this series will be on, I will find something on antigens and or antibodies rather, the 5 classes, and I want to talk about vaccination, etc.